Good morning, afternoon, everyone, and also extend my thanks and appreciation to Richard and Pia and InvestNME for the invitation to present at this meeting. The topic of today's talk, receptor and molecular pathway changes, perturbations identified in CFS-ME, and we're going to put up some data to provoke discussion about whether this illness may be a channelopathy, and in particular a calciopathy, or a calcium channelopathy. And we're using data from B cells and natural killer cells. I'd like to acknowledge my research partner, Sonia Marshall-Gradelsnick, and the others of the team who do all the work, Samantha Johnston, Tao Nguyen, and Tila Huth. You may remember last year we presented data on single nucleotide polymorphisms in the TRP receptors, the transient receptor potential ion channels, and that these TRPs have a likely significant role in changing how cellular signalling takes place. And in particular, let's look at natural killer cells. A lot of the, as you know, historical background of research in CFS-ME has been in natural killer cells and their impaired function. So we wanted to know what it may have been about the signalling that may cause such a change in function, and in particular the loss of the particular ability of NK cells to lyse or destroy target cells, which is their main job. Just briefly to recap the TRP family, there are 7 receptor families. There are numerous subtypes for each family with the exception of TRPA, which just has TRPA1. And there are numerous activating factors, physical and chemical factors, temperature and so on, and we'll talk more about that shortly. But just take a moment to look at the extraordinary array of factors that can impact and activate these TRP receptors, and it is their responsibility to negotiate these external signals with what happens inside the body. And in fact, they're located on every organ system and every cell in the body. And they're also known as threat receptors because all of those factors I pointed out a moment ago are threats to the human body. So TRPs actually act and are known as threat receptors, and they convert appropriate signalling into effector activity within the cellular metabolic pathways. This is it in a little more detail, and essentially what happens because of the TRP receptors being embedded in the cell membrane, they provide an amplification of signal through to second messengers such as calcium, and I'll be talking specifically more about calcium signalling in this talk, and how then that plays out to a very wide array of physiological functions. So we wanted to ask the first research question, did we find there was a significantly reduced NK cell lysis compared with healthy controls in this group? And the answer is yes, and you can see a significant reduction on the left in patients in their capacity to lyse the target cells. And this is standard finding from numerous laboratories previously. So the second question, were there any genetic changes in the isolated NK cells? And this is a summary of 14 or 15 SNPs, single nucleotide polymorphisms, from isolated NK cells. And these are all less than 0.05 in significance, the right-hand column. And the second from the right, the odds ratios are quite impressive with these. And of the 14 or 15 SNPs that we've identified here, at least 5 of those are TRPM3. So we became attracted to the idea that the TRPM3 receptor, the melanostatin 3 subtype, may have a significant role because of its high representation in the SNPs and the statistically significant SNPs that we found in NK cells. So we wanted to know more about the TRPM3, but to date the TRPM3 receptor had not been identified on NK cells or B cells. So we undertook that, and for the first time were able to report that yes, the TRPM3 is located on isolated B cells. These are CD19 positive B cells. And in the NK cells, the CD56 positive cells, both bright and dim subsets, have TRPM3 expressed. And these findings that I'm discussing today have been published just 2 days ago in a journal called Biological Research. So we then wanted to know, were there any differences between patients and healthy controls in terms of TRPM3 receptor expression in NK cells and B cells? And the answer was again yes, that there is a significant reduction, you can see in patients on the right, a significant reduction in TRPM3 receptors on B cells from CFS-ME patients. And also in a subset of the NK cells, the bright CD56 positive cells that are in the left, there is also quite marked changes in the dim CD56 cells, not significant but approaching significance, and they're shown there as well. So we then wanted to know, were the intracellular signalling pathways affected? And because these are so-called non-specific calcium channels, it was very important to then try to understand whether calcium signalling was substantially impaired. And calcium signalling is a very important part of intracellular mechanisms. People say, why is calcium so important? And to some extent, it's the Goldilocks of ions and ion channel movements, because some ions are too big, some ions are too small, calcium is just right, and it is able to bind into certain binding pockets, transmitter areas and so on, that enable various biochemical reactions to take place. Now this looks complex, but we'll walk through it. Now if you can see the top centre of the slide, that's where the receptors are located, and if you understand the concept, excuse me, that SNP-affected genes will potentially translate through to receptors which are damaged in some way, which means that there may be a gain of function, there could be loss of function, and their protein structure is potentially changed, and that in itself may be immunogenic. But the key point is that they allow calcium from outside the cell into the cell in response to these various signals. Now, it's very important that calcium is maintained outside the cell to maintain a certain electrostatic potential charge difference between the outside and the inside of the cell. In that way, very small puffs or signals of calcium within the cell can have a significant biochemical or metabolic effect. Just below that, you'll see that there is a large curved line. I can't tell you what's in that. We are currently producing about 9 or 10 further papers that discuss with some detail the changes happening within the cell. But we'll pick up the story here at protein kinase C. And what we have found, and I'll show you the data in a minute, but you will need to remember this slide to do so, that these MAP kinases, the mitogen activated protein kinases, are very critical in cellular function. And we have found increases in the P38 MAP kinase and the MEC1, MEC2 kinase. On the other hand, we found substantially impaired ERK1 and 2 kinases, and I'll get to that in a minute. So let's just deal with the left-hand side for the moment. So from protein kinase C effects, upregulation of P38 MAP kinase, which acts on a number of transcription factors in the nucleus. The short version of this is that there is an upregulation of largely inflammatory cytokines. So stringing that together, changes in calcium signalling, changes in PKC, upregulation of P38 MAP kinase, and an upregulation in interferon gamma, and tumour necrosis factor alpha, TNF-alpha. And you'll probably remember that these are the two main suspects in terms of how blood-brain barriers function. And blood-brain barriers tend not to like TNF-alpha in particular, as it causes an opening of those barriers. So if you can visualise this as the blood-brain barrier being something like mosquito net size, these cytokines have a detrimental effect effect on them and make them more like tennis netting size. So what that means is that extra molecules that are floating around in the circulation may be able to enter the brain parenchyma. So there's a hypothesis that may be worth working on in the future as to why the sustained inflammatory response that happens through the MAP kinase pathway is able to produce a sustained inflammatory response. Now remember, this is just in NK cells, in natural killer cells, but the same genetics, the same chemistry and so on should apply almost in every cell in the body to a greater or lesser extent. Moving now to the right-hand side, as I said before, and we've published this as well, I can't remember what journal that's in, but you just go to the NCNED website and we've got all the publications there, you'll be able to access them. So here there's a significant downregulation of ERK1 and 2. Now this is critically important because, as I mentioned before, the killer function of the NK cells relies on the cytotoxic vesicles here. And in those are perforins and granzymes which do the dirty work. So they're the ones that then target the invading bacterium or the cancer cell to cause lysis and then cause cell death, and that's how they do their job. But these black curved finger-like projections here are the actin cytoskeleton. Now, ERK1 and 2 and calcium are critically important for the way these vesicles migrate along the actin cytoskeleton, where they can get to the surface and then be triggered to do the task that they are to do. That's a very long story that may explain why there is impaired function of NK cells, which we saw at the beginning of the talk. So the next question is, what's happening to calcium within the cell? So the PhD students designed some very elegant experiments here where they wanted to know exactly what happened to TRPM3 and or the TRPM family and its capacity to function in bringing calcium into the cell. This is a lot more complex than I can describe here, but if you look at the CD56 bright cells under various forms of stimulation, that there's not a lot happening until you use substances called 2-APB and thapsigargan, which I think may come up, which are very specific calcium blockers, and they act in a number of very important ways. So they can block calcium at the level of the endoplasmic reticulum, and they can block calcium coming into the cell when exposed to different stimulation. And lo and behold, there is much lower calcium, statistically significant changes in the amount of calcium inside the cell. So that's a very, very kind of smoking gun suggestion that there is actually impaired calcium within the cell, and therefore there is impaired calcium signalling, because calcium is important in so many biochemical processes, which, as I say, I can't really comment on now. But just to show you the data that I mentioned before, the MEC1 and 2 and P38 substantially increased in the CFS-ME patients. And in the ERK1 and 2, it is significantly decreased. So by using those interventions of those calcium-targeted thapsigargan and 2-APB, that you're able to get a very good picture of how calcium is actually happening within the cell. So in summary, we found consistent reduction in NK cell lysis, as have other groups. We found significant genotype changes for the TRPM3 receptor, with at least 14 or 15 of those SNPs showing significant changes at less than 0.05 probability and quite high odds ratios, 4 or 5 or more, meaning that if you have that SNP, the odds of you having the illness are 4 or 5 times that of healthy controls. That we found a significant reduction. Not only did we identify TRPM3 on NK cells and B cells for the first time, and that's just been written up, that there was also a significant reduction in the TRPM3 receptors on NK cells and B cells in CFS-ME patients. The take-home message is that this is likely to result in significant changes in intracellular pathways, and that the reductions in calcium levels in the cytoplasm and the storage, a lot of storage of calcium inside the cell, inside the endoplasmic reticulum, is also compromised. So our conclusion is that impaired TRP receptor function and impaired calcium signalling and in stores is suggestive of a pathology for CFS-ME. Just as a sort of a footnote, the TRPM3 receptor, I'm not going to go into that in detail now, but just to go from the top down, these receptors are located in pancreatic beta cells. They likely have a significant role in perturbation of insulin and glucose regulation, and as we know, metabolic dysfunction is a key observation in ME/CFS patients. These are located on NK and B cells with impaired calcium as a consequence. The TRPM3 is actually located, as with all TRPs, on all body systems, but interestingly, because of their role as threat receptors, they tend to be in the part of the body that is going to be immediately exposed to any external toxin or whatever. And so the respiratory tree, digestive tract and so on tend to be associated more with these. And interestingly, they're also associated with pain signalling and nociception and the relay of pain signals, whether appropriately or inappropriately. And the generalised pain in the absence of recognised inflammation in CFS-ME is a very commonly reported and disabling symptom. TRPM3 also has a thermoregulatory role, so patients are often very intolerant of temperature changes, either temperature extremes, either high or low, and different TRP receptors identify different temperatures in the recording of that, and that certainly patients tend to be intolerant of those temperature changes. There's the team. I'd particularly like to acknowledge Mr. Ross Humphreys, the chair of our steering committee, who is here today. Mrs. Christine Hunter, AM, who's been a very strong patient advocate for many years. We have numerous clinicians that contribute to the research program. We're very grateful for the many patients who donate blood. We couldn't do this work without them. And these on the right, the people who do all the work, the PhD students, honours students, master's students and the postdocs. And I'd like to acknowledge our grants organisations. The Queensland Government has funded us from both sides of politics continuously for the last 8 years. The Mason Foundation, Edward P. Evans Foundation, the Alison Hunter Memorial Foundation and the Stafford Fox Medical Research Foundation. In thanking you, I'd like to, if you can see with the eye of faith, this is our research lab. We're the second top on this building, the health building at Griffith. You might see some white printing here that says Team ME at GE. Thank you very much.
Thanks very much, John. Thank you, Don. Now we're open for questions. Yes, two down here. There's a microphone coming in here.
I wanted to ask because many doctors have been finding calcium channel blockers like nimodipine very effective, and Professor Martin Paul suggested it might be because it blocks TRP receptors. So what do you think about this?
I'm not able to comment on the research of others, but it's certainly a fascinating topic. And
that's very diplomatic for you, Don.
Say again?
That's very diplomatic of you.
Thank you, Ian. I'm doing my best. That it's a very worthwhile area to pursue because the problem is that so many of these receptors work in opposite directions. And if this is the basis of the pathology or a contributing factor in the pathology, bear in mind there could be gain of function and loss of function. So picking exactly the right intervention is really going to be needle in a haystack, but it's a very good question to ask and an area of research that should be supported.
Okay. There was another one here, yes. Yes.
Many ME patients supplement, and in view of your findings, would it be more advisable to use to add calcium to your diet, or is it, in view of the channel problems, more advisable to withhold calcium?
Right. If I understood the question, is that appropriate to add calcium to the diet. Look, I really can't answer the question because we simply don't know enough about it. And in any event, the dysregulation, as I said a moment ago, could be gain of function, loss of function. So you'd really be guessing whether or not adding calcium may have an effect. So apart from that, look, again, a good question, but I don't have the answer.
I'd like to highlight the fact that amlodipine and fedipine can make many ME patients feel extremely unwell, extremely tired and forgetful.
Yes. So I think is mifedipine an antagonist of one of the TRP families used, I think, for pain relief, or it has other uses. And we'd actually looked a little bit about that as to whether clinical trials might be considered either in the function of that drug or in some way to counteract it. We're not at that point yet, but it's certainly a very interesting question, and we are aware of that effect. Thank you.
There's another one there too, Pia. Yes, thank you.
It's a question on behalf of a patient who is too ill to be here.
Yep.
She's been diagnosed as part of her problem, comorbidity, with severe osteoporosis. She's in her 40s.
Yep.
She's gone to a specialist for that aspect of her problem. She has been encouraged and chastised because she is reacting to the medications and the supplementation that she has been advised to take for her osteoporosis. What should she do, bearing in mind that she doesn't want to be seen as a vexatious and difficult patient, but she's tried and found it wanting, and she needs some good direction to take back to this specific clinician to say, we need caution here?
Look, I think you got the last part of that absolutely correct, that it is about discussing this with the clinician, saying that there are significant side effects and there has to be some different negotiation coming out of it. I mean, clearly I cannot make any comment about an individual clinical case, but when you do look at the potential for changes and perturbations and bad effects, side effects of things that happen to patients. I mean, don't get me started, but as you have, when you look at that list that we saw before of all the toxicants and external factors, exercise in particular, as you know in this forum we've said for a long time, patients should not be exercised. It should not be part of any sort of ostensible treatment regime, because what happens is you get, with exercise, changes and distortions of the cell membrane, and you get minor changes in osmotic pressure and so on, which can influence that, and that will cause exacerbation of the signalling disorder that underpins it in terms of this theory anyway. So with the clinic we have at home, I would say to patients, in terms of your physical activity, think of what you might think you're able to do tomorrow, but only do a third of it. And just do not push yourself beyond that, even when you think that you might be able to do it, because it will end in tears, because due to, we think, all of these perturbations of signalling, which have got multiple other effects. And as I said, I can't talk about some of that now, but stay tuned. These papers will come out over the next few months. You will get a clearer idea of the pathways which are dysregulated through this system here. That's all I can say, I'm sorry.
Yeah. In America, that would cost you $5,000 for that advice, or more. But I think talking to somebody at lunchtime, you know, you might get some further advice from other people in the room. Yes, Malcolm, and then we'll move on.
Just very quickly, most ME patients are very sensitive, ultra-sensitive to any kind of medication, and they become very easily distressed by it. There was a doctor, and I can't remember his name, in Edinburgh, who was giving an eighth of the dose of calcium blockers to introduce people into calcium blockers as a possible treatment for their ME. So very low dose, go in with very low doses. Don't start taking the normal dose.
Yeah. Okay, well, thank you very much, Don. Cheers.